So next thing that we're going to chat about, we're going to talk about tissues. Okay. So there's going to be a lot of flow charting here, because I'm a firm believer that when you have to basically memorize stuff, it helps to create some kind of a structure to that. Okay? There are 4 types of tissues. Epithelial, barrier, connective. Name is kind of speaking for itself. Muscular, movement. Well, actually not muscular, but more properly muscle tissue. And nervous. Well, also kind of self-explanatory, right? Communication and processing. Before we jump into each tissue separately, okay? And these are 4 types of tissues. When people say liver tissue, from the pedantic viewpoint, it's wrong. There's no liver tissue. There are multiple different tissues in the liver. There's connective, there's epithelial, there are muscles in the liver, like smooth muscle in the blood vessels and all that kind of stuff, okay? Now, a few things about the tissue development. 2 terms that I'm going to be using in this course and other professors are using in other courses. It's a term, proliferation, which basically means reproduction. So when cells proliferate, they become more numerous. Okay, can you excuse me for a second? And then the next term is differentiation. Which means specialization. When cells differentiate, they become more specialized. And it's the only, when we say B cells differentiate into plasma cells, it means that they become more specialized. Okay, we're good? So all of this explains the very, very basic of embryonic development of the tissues. So at the level of the embryo, we start with it's not even an embryo yet, really. We start with totipotent stem cells. The term totipotent means potent can be toti, total, everything. So at this point, we're talking actually not so much about embryo, we're talking about blastocyst. So, like few days after conception, when it basically crawled out of the fallopian tube and attached to the wall of uterus. Okay? That makes sense? All cells are the same, basically. Now, totipotent stem cells differentiate, that's your differentiation, into pluripotent stem cells. So they're already a little bit specialized, and within pluripotent stem cells, we identify 3 types of embryonic tissue. Ectoderm, that's the outer layer, the most outside, basically that's what we're talking about. Ectoderm those cells give rise to organs such as nervous system and skin. The innermost layer, which is called endoderm, gives rise to systems like respiratory or digestive. And in the middle is mesoderm, which produces such interesting, well, muscles are not so surprising, but kidneys maybe a little bit are. Does that make sense? This whole layout here. Now, you may wonder, what about the examples? So I'm a firm believer that, I should not tell you, know all the organ systems that are derived from mesoderm. It's just too much. It's not justified. So those are examples that I expect you to know. Does that make sense? Like, I can ask you, which of the following organs is derived from endoderm? And it's going to be either respiratory or digestive. It's not going to be, anything else that's also derived from endoderm that we did not say in class. Am I clear? I'm trying to kind of emphasize this idea. Now, pluripotent stem cells can specialize even further. For instance, not for the exam, bone marrow is the mesoderm product. Okay? So in bone marrow, you're going to have what's called multipotent stem cells. Let's say you have bone marrow stem cells that can become red blood cells, T lymphocytes, B lymphocytes, macrophages, neutrophils, all kinds of blood cells. Do they have many capacities? Yes. Are they limited? Yes. When multipotent stem cells, sometimes they differentiate even more, and they become what is called unipotent stem cells. In some way, for instance, a cell called monocyte, you can consider it a unipotent stem cell because it can differentiate further into macrophage. Okay? It has only one capacity. Keratinocytes in the basal layer of your skin are stem cells. They divide and generate non-dividing keratinocytes. You have stem cells in your nose that differentiate into olfactory neurons. This is why when you inhale something toxic and kill off your olfactory epithelium, it gets restored eventually. Does that make sense? And eventually those unipotent stem cells will become, and I put this dashed line, meaning that they can be multiple steps. It's not super important. They become terminally differentiated cells. Okay, so interestingly, the fibroblasts that are found in your skin, technically speaking, are not terminally differentiated. You can differentiate them in vitro, in the lab, into fat cells. That's it. Fat cell doesn't go anywhere else. Okay, we good? Great. There's one topic that I cover in lectures pretty extensively. It's stem cell research. Well, more extensively than here. So, obviously, there is only one reliable source of stem cells now. Those are embryo. I'm not going to touch on the ethical component here. Forget about it. So we can't work with human embryos in the United States. So let's pass on that. But in 2012, there was a Nobel Prize awarded two American, well, one of them Japanese actually, two American scientists, but works in the University of California, San Francisco. It was awarded for the development of what's called inducible pluripotent stem cells. So what you do, you take either this or this, doesn't really matter, and you go Benjamin Button on them. It's called de-differentiation. It's kind of there, the spelling. So you have inducible, that's letter I, pluripotent stem cells. Okay? So you can make this. And then you can use those inducible pluripotent stem cells, technically you can, to grow tissues and organs of your choice. It's not that simple, because to my knowledge there are no actual approved medical treatments so far exist. So like we can't grow a kidney or a liver. Well, we probably can, but it's very complicated and it's not been tried in human patients. The research has been quite extensive on animals actually. The advantage, well, obviously, first of all, you can grow whatever, and that means that you don't need to wait for an organ donor, and you don't need to wait for a match, right? And if, for instance, there is a match, and they're suitable, there's still going to be difference, there's still going to be rejection. In this case, there's no rejection. Because this, like imagine hypothetically, you take patient's cells, fibroblasts, de-differentiate them, and grow a new kidney, and put it in the patient. Those are patient cells. They aren't foreign for his immune system, or her immune system. Does that make sense? So the rejection problem is solved. Unfortunately, well, but we're not there yet. Okay? Questions here? Okay, we're going to start with epithelial cells, epithelial tissue actually. What I want you to, well, what a general property is. It is a barrier. So schematic picture of the epithelium can be like this. That's probably the apex of my drawing capacities. So these are epithelial cells. Here on this side is lumen. So what's a lumen? Can you imagine a pipe? Just a pipe. What's inside the pipe? Like pipe. I will go to Home Depot and grab a PVC pipe. What's inside of it? Nothing. That is a lumen. Whatever is inside a pipe is a lumen. Whatever's inside a bag or a bottle, you can call this, this space is a lumen. So a lumen of that bottle is filled with water right now. Does that make sense? This is the proper anatomical term that's used for like a lumen of a stomach, a lumen of a trachea, a lumen of a of the intestine, and stuff like that. A lumen of a blood vessel. Clear? On the other side, tissues. Okay, so think about it. Think about a blood vessel. It's a good example. Inside a lumen of the blood vessel is what? Blood, right? What's on the other side of epithelium? A whole bunch of connective tissues, can be muscle tissues, whatever is this blood vessel is going through. Make sense? This is how, like when you're going to be looking at the slides today, how do you know where epithelium is? It should bound the lumen. Lumen can be enclosed if you do the cross section, or it can be just empty space. If you do longitudinal, so if you cut a PVC pipe along, that's going to be like just nothingness, okay? So, let's move on. These cells are attached to the basement membrane. Okay? It's really important. A lot of physiological processes, a lot of pathologies are due to problems with the basement membrane in the epithelium. Okay? Think of it this way. Those are bricks, that's the foundation. Okay? Good, clear? And the sides of epithelium have specific names. So that side, the side that faces the lumen, is apical. This side, the side that faces the basement membrane, is basal. Why this is important? Apical and basal sides of epithelium may, and often do, different properties. For instance, the set of transmembrane proteins found on the apical side of your intestinal epithelium, that set is totally different from what's found on the basal side. You see what I'm trying to tell you? Like in your garage, you have a garage door in front, and you probably have a small back door in the back. So it's not like you just blasting through, you know. Make sense? Very important feature of the epithelial tissue. It is not vascularized. It means that if you look at any epithelial layer, there are no blood vessels in the layer. Make sense to you? So you may wonder, what about nutrients? What about oxygen? There are blood vessels adjacent to the epithelium, so diffusion. Just simple diffusion. Okay? So far, good? Let's get to the classification. Now you need to know specifically 6 epithelial types, what they function, and get a decent idea where they can be found. Again, I told you before, I'm not here to trick you, so I'm not going to ask super specific questions like what kind of epithelium is found in the lactiferous ducts in the mammary glands. It's not a point, okay? It's just for you to understand. So you can classify epithelium based on the number of layers. If you have one layer, the epithelium is simple. Okay? If you have more than one layer, epithelium is stratified. Okay? Meaning that there are stratas, layers. Okay? Does that make sense? Now, second approach to classification is the shape of the cells. So cells can be very flat, even more flat than these ones. Squeeze them like 3 times. That's going to be squamous. Okay, now look, you've got simple squamous epithelium. So you have, check this out, one layer of very flat cells. One layer of very thin flat cells definitely does not make for a good barrier. So it probably won't prevent the transport. But it will allow you to partition, okay, the locations where you have transport, diffusion, or filtration. Not filtrations, filtration. Where you're going to have it? Transport in your blood vessels, transport across the vessel wall. In your lungs, respiratory gases will diffuse across the simple squamous epithelium. Filtration in some parts of your kidney, in the glomerulus, okay, of each nephron, you have filtration of the blood. Does that make sense, the whole idea? Good? Then you've got cube-shaped cells. So that makes for cuboidal epithelium. The main function of cuboidal epithelium is secretion, and in specific locations, absorption. So if cuboidal epithelium can secrete, it's going to be found in the secretory organs, right? We're talking about various glands. Many glands contain cuboidal epithelial cells, like thyroid and some parts of the pancreas. And absorption. Absorption, more specifically, kind of reserved for the kidneys here. Okay? That makes sense? And the third sort of a proper type is kilometer. Cells shaped like a column. Okay? And here function is also secretion and absorption. Now, let me resolve some confusion here. I cannot imagine, by any means, that you will know, like by tomorrow, all the places like that cuboidal cells do secretion in thyroid gland and kidney tubules, but columnar cells do secretion in the intestine. You know, I can't expect this. So if I ask you which of the following cell types does a lot of secretion and absorption, I'm not going to throw columnar and cuboidal at you at the same time, obviously. Because you're in your right, you say, oh, I don't know which one do you want. Okay. Now, columnar, they are mostly not in the kidney, not in the glands, some in fallopian tubes, some in bronchi, digestive tract, starting from stomach, entirely columnar. Stomach and the entire intestine, columnar epithelium. That makes sense? They do secrete, very good at mucus secretion, like phenomenally good. Stratified. Only one stratified type. The proper stratified type is stratified squamous epithelium. So you have many layers of thin flat cells, but many layers, but thin. So obviously you can easily lose outside layers, which means this protects against abrasion forces. Does that make sense to you? Right here, your skin. Actually, it's very easy to figure out what are the body parts that experience abrasion except your skin. Do you eat? Do you eat like normal people's food? Like nuts? Your mouth. There's a lot of abrasion going on. Your gums are covered by stratified squamous epithelium. Interestingly enough, your nose is not. Let's not pretend that we're not picking the noses. Do you do this? This. Your eyes. There are two places that students very rarely think about. A lot of abrasion going on. That's skin. Students don't talk about them because those anatomical locations are not considered to be worth mentioning in good company. Huh? Not entire, but yes. Vagina and the glans penis, yes. anus. A hell of a breccia. No, seriously, there is a boundary. There is a boundary. Because it's a line, pectinate line, that separates when kilometer epithelium ends and stratified squamous begins. That makes sense? Specifically anal orifice. Another esophagus also. It's basically like your upper part of digestive system. So we're talking mouth, oropharynx, laryngopharynx, esophagus. That's because food is still pretty rough up there. Okay? We're good so far? There are two types that fall under the umbrella of these, but they're kind of weird. So one of them, I'm going to put this under the simple pseudo-stratified kilometer. Okay?
And this is a different tissue?
Say again?
And this is a different
Yes, yes, it is simple epithelium, but we call it pseudo-stratified because it looks like there are multiple layers. You follow me?
Yes.
But they're not. And it looks like because cells are weirdly shaped, and you can see 2, 3, sometimes 4 kind of lines of nuclei under the microscope. And these 4 lines they look like 4 layers, but they're actually not. Does that make sense?
Could you spell the first 3 letters?
P-S-E-U-D-O. Sure. Sorry for the chicken scratch. And here, under the stratified epithelium, transitional. The interesting feature of transitional epithelium, and it's found exclusively in the urinary system, it's stretchable. So only ureters, urinary bladder, urethra. So you have many layers. When they're not stretched, they look cuboidal. You stretch them, the cells transform into squamous. You relieve the stretch, go back to cuboidal. Does that make sense? Good. So these are 6 types that I can ask you about, okay? More functionally, okay? So I deliberately put functions here. So how these cells are held together, I mean these cells. There are so-called junctions. Okay. 3 types of junctions that I want you to know. Gap, tight, and adherent. So let's clear it out. What is the gap junction? Imagine that this room is a cell, okay? And look there. I don't step out of the count, that's the only reason I would walk there, otherwise. And that hallway is the cell too, different cell. The door is a gap junction. So basically it's ion channels that connect 2 adjacent cells. Are you following me? When conditions in one cell change, the conditions in another cell immediately change. So what happens, think of it as like a rumor spreading through the cells. You see what I'm saying? So you can have thousands and thousands of cells connected to each other by gap junction. You can send a signal to one cell. It's going to change, and these changes through gap junctions will be transmitted like a domino effect to all other cells. So these cells will act together It's a feature of smooth muscle cells, especially cardiac cells to some extent. Tight junctions. These are two epithelial cells. You see how tight they are? Nothing can get between them. This is the great barrier. For instance, blood vessels in your brain, the epithelial cells that cover them from the inside are connected to each other by tight junctions, because the last thing you want is some kind of a bug to migrate from blood vessel into the brain, like squeeze between. Does that make sense? So tight junctions prevent kind of leakage. Adherent junctions, just a garden variety. They hold cells not too tight. You have some space, as you can see on this drawing, so some stuff can get through. Not all of it, usually kind of smallish stuff. So like proteins usually can't, but small molecules can. You following me? That's in adherent junctions. You probably see junction adherents, desmosomes, hemidesmosomes. I'm not going to ask those particular specifics. Okay? Basically, adherents allow them to stretch a bit. Desmosomes, hemidesmosomes just hold. Okay. Questions so far? Now, last bit about epithelium. Epithelium forms secretory glands. And secretory glands can be divided into two major categories. Endocrine glands that secrete into blood, and more broadly extracellular fluid. We're going to study them. You're going to study them. I don't know if you're going to take EP2 with me or not. You're going to study them in the endocrine system, right? So examples would be thyroid, pituitary gland, ovaries, testes. Obviously, for ovaries and testes, not the only function, right? Like adrenal glands, they secrete, they release hormones, and that's important. Hormones into the blood. Exocrine glands do not secrete into the blood. They're still inside. So for instance, look, you have pancreas, which is both endocrine and exocrine gland, but its exocrine component, it produces pancreatic juice that gets into your intestine. Your liver is a gigantic exocrine gland that produces bile. Does that make sense? Your intestines are covered with rather small intestinal glands or crypts that produce into the intestine. Sweat glands are exocrine. Now, exocrine glands can be divided by the way of secretion, how they do it, in three categories. Merocrine, apocrine, and holocrine. So let me explain, kind of animate them, what the difference. So, Katie, question?
Yeah, what's the first one?
Merocrine, M-E-R-O-C-R-I-N-E. So merocrine gland. Let's use it. Let's use the glands that are in your skin, and we simultaneously going to cover skin glands. When you work in the heat, you produce copious amounts of sweat, right? On your forehead, on your anterior or posterior trunk, you produce it to cool yourself off, right? This sweat is just a little bit salty. It's water and a little bit of sodium chloride. Those are merocrine glands. What they do, they package that sweat really nicely into little vesicles and release it and the cell is unchanged. You see what I'm trying to say? Like, cell is not damaged by any means. Now, you do have so-called Epicreen sweat glands that are located in your armpits and in your groin. The smelly places, right? Why are they smelly? Because in Epicreen secretion, imagine that I'm that secreting cell. I'm not just spitting some stuff. My arm gets chopped off. So the part of the cell with everything in it gets secreted. Proteins, fats, you name it. You see what I'm saying? So all that really juicy cocktail with cytoplasm and all cytoplasmic components gets on your skin, and there are bacteria that are ready to pick it up and digest it, and they produce the smell. Make sense? And finally, Holocrine secretion. The entire cell explodes, basically. Well, not explodes, it dies. Dies, disassembles, and all cellular contents are released. The holocrine secretion is characteristic for sebaceous glands that produce sebum, the oily substance that lubricates your skin. Does that make sense, Tim?
Can you repeat that, what you just said?
Yes. So holocrine glands, these cells, they die. Every time they secrete, they just blow up. And all the cellular content gets released. That's the definition of holocrine secretion. So for example, sebaceous glands found in your skin, they secrete by these mechanisms.
Can you repeat the middle one?
The middle?
Yeah.
So a part of the cell gets chopped off. Okay? Basically. I mean, cell is still alive. It will regrow that part, but loses. The important part, everything that's in that chunk gets secreted. Okay, good? Questions? That's going to be a long video because that's going to be all tissues. So we're climbing on a mountain. Because the mountain, any questions here? Because the mountain here is the connective tissue. Not because it's complicated, just the highest number among all the tissue. Like it has a lot of different types, but they can be very, very nicely structured. Before we structure, okay, we need to discuss the components. So connective tissue consists of the matrix and cells. As you can immediately say, matrix is not cellular, right? I mean, matrix is molecules, proteins, proteoglycans, protein structures, but not cellular. So matrix is a ground substance a goo that consists of carbohydrates, huge carbohydrate polymers, proteoglycans, and so on and so forth, and protein fibers. Now, in the protein fibers, there are three types here: collagen fibers, elastic fibers, and reticular fibers. The physiological properties of a connective tissue are largely determined by which fibers are most dominant. You have more collagen, more mechanical resistance. Does that make sense? If you increase the number of elastic fibers, more elasticity. If you increase the number of reticular fibers, the tissue will have better capacity to filter and contain stuff. You follow me? Now, cells. Now, we've got several here. So first and foremost, we've got fibroblasts. So that's the main, again, we're talking about a connective tissue. Does that make sense? So there are some more specialized connective tissues that will contain very special cells, not fibroblasts. They're going to be named slightly differently. So what fibroblasts do, they make matrix. They produce matrix. Then you have mesenchymal cells. Those are stem cells. They differentiate into fibroblasts. Makes sense? It's just unipotent stem cell that divides and differentiates into fibroblasts and so on and so forth. Okay. And last but not least, connective tissue is pretty rich with white blood cells. You'd be like, wait a minute, aren't they supposed to be in the blood? Some of them are, some of them are not. Some of them are sticking around in the connective tissue. Does that part make sense? Good? Awesome. So let's do a flow chart. And that lays, I did that multiple times, it works great. So connective tissue can be divided into two giant categories. One is connective tissue proper, like classic, okay? And another is specialized. Okay? In the specialized connective tissue, we can identify blood, bone, and cartilage, to which I return a bit later. Let's just get the easy stuff out of the way. So blood, right? You understand, right? Where the blood is and what it does. I don't need to, like. And bone, like, hello Benjamin, right? So that's bone. It's all connective tissue, very specialized, very special cells. For instance, what's matrix in the blood? Plasma. Does it have those proteins? No, it doesn't. It has albumin, it has beta globins, gamma globulins, but that's a very, very specialized connective tissue. Does it have all those cells? No. It has white blood cells, red blood cells, platelets, but you know. See what I'm saying? Let's start with the proper one. So, proper connective tissue can be loose. And in the loose, there are three types. First one is areolar. This is kind of a jack of all trades. It cushions virtually all the organs. Okay? You cut open a stomach, gut, liver, kidney as a whole, as the whole organ, you're going to find areolar tissue surrounding it, providing that nice cushioning. Okay? It also contains a little bit of itch, a little bit of collagen fibers, a little bit of elastic, so it can stretch, a little bit of reticular. So if pathogen comes through, it won't be that easy. Does that make sense? And then you have adipose tissue. Energy storage, that's fat. I mean, I greatly downplay the importance of adipose tissue. It plays an immense role in inflammation, really important, really fascinating stuff. Reticular. So reticular, I'm going to put this way, lymphatic filtration. Okay? So what's lymphatic filtration? If you think about your body as the house, okay, you can think about cardiovascular system as the water pump. You see what I'm saying? You have a heart that pumps, and all the pipes are blood vessels. Make sense? But once you took a shower, all the water that you used goes where? To the sewage, right? That's your lymphatic system. It's a sewage. And what do you want to do with the sewage? You want to filter it and contain all the crap that's in the sewage so it doesn't get back to your water supply, back to your blood. Does that make sense? And that's what reticular fibers in the reticular loose connective tissue that's found in the spleen and lymph nodes, that's what they do. Good? Okay, cool. Now, once we have loose, we gotta have dense. Also 3 types. Regular, dense regular connective tissue. So why this tissue is dense? Because it has a lot of collagen fibers. Make sense? Now, in dense regular connective tissue, collagen fibers are nicely aligned like this. So it can withstand a substantial tension. It's not real good at forces that come at an angle, but if you pull in one direction that matches the direction of collagen fibers, you're going to be fine. So it will comprise structures like ligaments and tendons. Well, if you have regular, you've got to have irregular. And in this case, the fibers will be in kind of, all freaky, weird directions. And what it makes this tissue suitable for, it can resist tension that's applied in different directions, at an angle. You following me? So, for instance, think of like Achilles tendon. If you look at any of the Arnolds right there, you can see a giant white patch that goes from the calf muscle all the way down to the heel. The white, sort of long triangular patch, that's the Achilles tendon. The way you pull on it is, like vertically, right? That's the tendon, but on your skin you pull in all different directions. So you're going to find dense irregular connective tissue in the skin, as well as joint capsules that experience forces of various kinds. And finally, I have no place for cartilage, so we're going to pull it down. We're going to stick this one down here. It's dense elastic connective tissue. The term is kind of self-explanatory, right? So it can stretch and recoil. So we're talking about arteries and bronchi, especially large ones. So your large arteries, they called elastic actually, like aorta or subclavian. They can stretch quite dramatically and they can recoil back. Your bronchi can stretch and recoil. You see, this is why. Dense elastic connective tissue. We good? Now what about cartilage? 3 types. Now for these types, I do ask you to know where they are. This is real easy. Do I need to explain what elastic cartilage can do? Obviously, it's elastic. Found in 3 places in the human body. Place number 1. Place number 2. So your external ears. And, well, I'm not going to rip it out, but here in the larynx, you have a flap, a valve, called epiglottis. That's elastic cartilage. That's it. Nowhere else. Fibrous cartilage has a ton of collagen fibers, a lot of them, which means it's very mechanically resistant and kind of, stiff. Where are you going to find it? You see those intervertebral discs here? They all experience compression, right? So that's fibrous cartilage. Do you see this pubic symphysis here between the coxal bones? These bones come at each other. So also elastic cartilage. No, not elastic fibers, sorry. And something that's not shown, but here you're going to have 2 menisci, the medial and lateral menisci. So menisci in your knees that experience the compression, also fibrous cartilage. Questions so far? What about all other cartilage? It's going to be hyaline. It's most common. Interestingly enough, you know what the name means? The name hyaline means shiny. That's all. Like, doesn't explain the function at all. For instance, on Benjamin, this hyaline, the costal cartilage is hyaline cartilage. That's hyaline cartilage. Ends of the bones are covered in hyaline cartilage. The part of your nasal septum right here, the one that's kind of bendable, that's hyaline cartilage. Question so far? That's your connective. We have 2 tissues. We're going to do skin afterwards. It's going to be a longer stretch. And then we're going to take a short break and go to microscopes. Okay. Muscle tissues. Three types, pretty straightforward. We're going to start with the skeletal. So what are defining features of the skeletal muscle? It's the only muscle type that can be voluntarily contracted. Voluntarily controlled, I'm sorry. Voluntary control, okay? The cells are straight, non-branched, and they are striated. This really helps in identifying muscle tissue. If you look at the cell, it's really, really long, okay? And it has stripes that go across. Cells are polynucleated. Okay, and let's see. Oh yeah, the stimulus. Nervous stimulus. What does that mean? In order to contract a skeletal muscle, the signal from the brain should be sent. In the overwhelming majority of cases, the signals are voluntary signals. Does that make sense? And this kind of voluntary nervous stimulus, this is why they go together. Because that is the only system that has consciousness of sorts. Does that make sense? You don't want your skeletal muscles to start contracting in response to hormones or chemicals. Like inject someone with stuff and people's moving around. Does that make sense? Without mind control. Cardiac. Speaking of skeletal, do I need to explain where it can be found? Or you can just look at all those beautiful models and just figure it out? That's skeletal muscle, okay? Cardiac, involuntary, found only in the heart. Branched cells, striated. It's kind of a shame, I have to say, that they striated at this point. You have no idea why. And I'm definitely not going to go into why. The only thing I'm going to say, it's not for this exam. The similarity in structure implies the similarity in function. Does that make sense? You see, structure determines function and vice versa. So the mechanism of contraction for skeletal and cardiac muscle is the same. They're structurally very similar, actually. 1 to 2 nuclei, mostly one. And stimulus can be nervous, or hormonal, or chemical, or even mechanical. You can't make cardiomyocytes contracting by mechanical stimulation. So far we clear? And the third type, the smooth muscle. Only involuntary. Okay. Cells are spindle-shaped. Non-striated, which you should think at this point, so the mechanism is different, right? Uninucleated. So one nucleus. And the stimulus, same as cardiac. Does that part make sense? Okay. Questions here? I mean, we have one tissue left, which is, I told you, connective was a summit. We went over the summit, and now we descend into the valley. Nervous. Two cell types. You have glial cells. Glia, glial cells, neuroglia. The function broadly is support. Since next, terrifyingly, 3 units will be about nervous system in one or other instance. We're going to have a chance to talk a lot about this, so I don't spend too much time. Glia and neurons. All kinds of stuff, so communication, response. One of analogies that I use every time I talk about it, I compare numbers-wise and sort of function-wise. So this is Guns N' Roses, you know, in the golden team, McKagan / Axl Rose, Matt Sorum, Dizzy Reed, and so on. There were 6 of them. You know, okay, including, okay, let's say 9, with female characters, so on and so forth. So let's say 9. And then you have stage crew. Everybody from personal therapist to a guy who assembles the drums for Matt Sorum. That makes sense? You're going to have like 100 of these folks. The ratio of glial cells to neurons is 9:1. So 90% glial cells, 10% neurons. Neurons, without neurons, you have no Guns N' Roses. Like, you have no brain. But without glial cells, they can't play. There's going to be no gig. Does that make sense to you? So that's it. Questions on the nervous? Okay, awesome.